Welcome to the MOFLUX site at the University of Missouri's Basket Wildlife Research and Education Center, just east of Ashland. This video will take you on a virtual tour of the MOFLUX Ecosystem Observatory to show you how we are monitoring the forest from roots to shoots and leaf to landscape. The tower that you see here is the focal point of research activities and is equipped to measure the breathing of the forest. In other words, how much carbon dioxide is breathed in and out and how much water vapor is breathed out. My name is Dr. Jeffrey Wood, and I'm an assistant research professor in the School of Natural Resources at the University of Missouri. I'm one of the principal investigators at the MOFLUX site, and I'll be your tour guide today. The MOFLUX Tower is the centerpiece of a long-term research project initiated in 2004 by Oak Ridge National Laboratory and the University of Missouri, and funded by the U.S. Department of Energy Office of Science, Biological and Environmental Research Program. The overall focus of the research is on understanding how extreme events and droughts impact forests. We use an integrated research approach that combines measurements enabled by advanced instrumentation with modeling to study detailed processes like leaf-level photosynthesis in great detail, as well as whole ecosystem behavior in terms of the cycling of carbon dioxide and water vapor between the forest and the atmosphere. Our fundamental research improves scientific understanding of how plants function and respond to environmental conditions to improve ecological and climate forecasting. This will allow us to answer questions like how will the forest change over time and how healthy will the forest be in the future. Because we do basic scientific research, we generate knowledge that can be applied to other areas of inquiry and eventually commercial applications. You can follow me now as we go see the site in more detail. The MOFLUX site is situated in an oak hickory forest that is about 85 years old, where white oak is the dominant tree species and accounts for about 40% of the basal area. As is common throughout much of the eastern deciduous forest, we find a lot of maple trees in the understory and limited oak regeneration. We're now at the base of the MOFLUX Tower, and immediately adjacent to us is our climate-controlled support building where we house instruments and equipment needed to support environmental sensing outside. The tower is the centerpiece of the research infrastructure and is situated at the center of the closed canopy forest site. We have various instrumentation deployed in the nearby forest, as well as 24 vegetation plots distributed along transects that radiate out from the tower. The tower is 105 feet tall and is equipped with a variety of sensors that allow us to measure the flows of carbon dioxide, water, and energy into and out of the forest along with the weather. To measure these flows of carbon dioxide and energy, or what we refer to as net ecosystem exchanges of gases and heat, we use a technique called eddy covariance. Here is a miniature model representing our tower with the eddy covariance system out on the end of this boom. You can visualize how the method works best by imagining that we drop an imaginary box on our forest. To calculate the net ecosystem exchanges, we need to know mass or heat flow across the top of the box and the change of concentration within our box. Technically, we also need to know the horizontal mass and heat flow through the box from side to side, but these are often in balance and we can ignore them. The mass and heat flow across the top of the box is estimated from very fast measurements of wind velocity, gas concentrations, and temperature that are made at the top of the tower. You can see our main instrument boom on which these sensors are mounted here. These measurements are made continuously at a rate of 10 times per second. These fast measurements actually resolve the transport of gases and heat by eddies that are swirling around in the atmosphere. Although you cannot see the eddies, they are everywhere in the atmosphere, particularly near the surface. These eddies are the main way that forests interact with the atmosphere and are able to breathe in carbon dioxide. To estimate the change of concentration in our conceptual box over time, we measure profiles of gas concentrations and temperature from the soil surface to tower top. We measure carbon dioxide and water vapor concentrations at 12 levels and temperature at 8 levels. You can see the radiation shields in which temperature probes are mounted on this leg of the tower. Once we know the vertical transport across the top of the box, and the change of concentration inside the box, we add them together to calculate the net ecosystem exchange. We're now standing at the top of the tower and looking in a southerly direction. Below us and to the right, you can see our main instrument boom extending from the tower, and our eddy covariance system is deployed on the end of the boom. The sensor that looks like two claws on the end of the boom is a sonic anemometer. It measures the wind velocities in north-south, east-west, and vertical directions, as well as the air temperature. The white cylinder is a gas analyzer that measures carbon dioxide and water vapor concentrations. These instruments make the measurements that allow us to calculate the flows of carbon dioxide, water vapor, and heat across the top of our imaginary box. The sensors sticking out to the sides measure incoming and outgoing radiation for 3 different wavelength ranges. For example, the sensors on the right measure solar radiation and long-wave radiation. The second boom that you see to the left is deploying components of novel instrumentation that we use to measure something called solar-induced chlorophyll fluorescence, or SIF for short. SIF science is an emerging approach allowing for unprecedented remote monitoring of ecosystem-scale photosynthesis. When plants are growing and photosynthesizing, a small amount of the solar energy that is absorbed by the leaves is not used to drive photosynthesis and is actually emitted as SIF. In other words, the leaves are glowing. We just can't see it with our own eyes. Luckily, our instruments can. Our system here, which we call the Fluorescence Automated Measurement Equipment, or FAME for short, was custom designed and built at Oak Ridge National Laboratory. The rotating piece on the end of the boom is a fiber optic cable that alternately views the sky and the forest canopy to sample the incoming and outgoing radiation, respectively. The light passes through the fiber optic to a spectrometer housed in this climate-controlled enclosure to make the actual measurements that we use to calculate SIF. This is our prototype system, which has undergone several iterative improvements to a point where it has now been licensed to a manufacturer in the private sector. We're going to climb down now, and then I'll show you the inside of the instrument building and explain how we measure the profiles of carbon dioxide and water vapor. The instrument building is a secure location where we mount active instrumentation, store other equipment, and have an air-conditioned space for respite on the hottest days of summer. Follow me inside and I'll show you our profile measurement system. Remember, this is how we measure the concentration change inside the box. This metal cabinet houses the profile monitoring system. On the front door here, you can see the analyzer that measures carbon dioxide and water vapor concentrations. This analyzer is always sampling air from one of the 12 intakes that are positioned at different levels through the depth of the canopy. Our sampling system inside the cabinet automatically switches the height that is being measured every minute sequentially from bottom to top. So a full cycle takes 12 minutes. Here is the profile system. As you can see, there are many different components. This is the data logger. The data logger is the brain of this system, and it can control systems as well as record data. For instance, the data logger controls these relays, which in turn control the valves on these manifolds. If you look at these relays, the active valve is indicated by the red LED. When it changes, that signals a switch to the next valve. All of the equipment that you've seen today on the tower and in the shed run continuously 365 days a year. This generates a huge amount of data, and we take that huge data set and summarize it into data products that represent half-hour intervals of time. We're now going to head out into the forest, but before we do, I'd like to review a couple of points. First, what you've seen so far is the equipment that we use to measure the breathing of the forest. That's the flows of carbon dioxide, water vapor, and heat into and out of the forest, along with supporting weather variables that help us to interpret that breathing of the forest. Second, with these measurements, we can look at how rapid changes in environmental conditions affect the breathing of the forest, for example, over the course of a daily cycle. Or we can also look at accumulated effects over a longer period of time, say over the course of a growing season, and investigate how soil drying or drought affects the carbon dioxide balance. Remember, what you've seen so far is how we measure whole ecosystem behavior. But we also want to know and understand the underlying processes that give rise to that whole ecosystem behavior. So now we're going to go out into the forest and see how we measure sub-ecosystem processes. The two that we'll investigate are soil respiration and root growth. We are now in the middle of a defined soil monitoring area. We have equipment to measure how much carbon dioxide is emitted from the soils. This carbon dioxide can be from either the respiration of microbes growing in the soil or from the activity of living plant roots. We make these measurements because we're very interested in how above-ground parts of the forest are connected to the below-ground parts of the forest. The soil respiration measurement system consists of 16 chambers that you can see distributed around the area in these two boxes. One box is a gas analyzer and the other the sampling system. The boxes work together with the sampling chambers to form a complete sampling system. Every few minutes, it automatically selects a chamber to activate and makes a measurement. A chamber measurement involves the lid closing for a couple of minutes, during which time air is circulated in a closed loop between the chamber and the analyzer. Over time, the carbon dioxide concentration builds up inside the chamber. From this accumulation rate, we calculate the carbon dioxide emission. It takes a little less than an hour to collect a full sequence of measurements from all of the chambers. The system will then get a break for a few minutes before it continues on with the next hour's measurements. We also peer beneath the soil surface to look at living roots. We look below the surface using mini rhizotrons, and you can see these PVC pipes sticking out of the soil around the area. This one has the mini rhizotron camera inserted into it right now. Inside the PVC tube is a second clear plastic tube, and that's actually what the camera slides into to take pictures. Each week, someone comes out to take pictures of the roots in each of the mini rhizotron tubes. You can see the whole imaging system shown here, the camera, and the computer. The camera is on the end of this stick, and there are indexed locations, and a picture is taken at each one of those indexed locations as it is withdrawn from the tube. Those pictures can then be stitched together to image the roots along the path. We have one final stop on our tour, and we'll head there now to see one of our vegetation plots. So follow me. We're now standing on one of our vegetation plots. These plots are crucial to understanding structural and compositional changes of the forest through time. On each plot, every tree has a unique ID tag. During the growing seasons, we check the plots to see if trees have been damaged by storms or have died. With these data, we can track stand dynamics and things like tree mortality in relation to drought. In addition to the tree inventory, we also collect leaf and woody litter in these tubs for which the collection area is known. The litter fall is important to know because it represents an important source of carbon for the soil, which feeds the microbes. This vegetation monitoring represents some of the work that involves boots on the ground and people power as opposed to the sophisticated instrumentation that you saw earlier. We make many other ecological and physiological measurements, and you can learn about some of those in the other videos. I hope that you've enjoyed your visit to MOFLUX today and have learned how we work to study the breathing of the forest. I also hope that you've gained an appreciation for our interdisciplinary research approach and how we strive to understand both whole ecosystem behavior as well as the underlying processes that give rise to that behavior. The MOFLUX Tower that you've seen in this video is part of both national and global networks of similar sites making common measurements in a broad range of ecosystems and climate. Such networks make it easy to compare and contrast across ecosystems and climate. The length of data records at these sites is ever growing, and there are many sites with 15 to 20 years of data. These long-term data records are crucial because it makes it possible to understand how ecosystems respond to dynamic and variable environmental conditions that include drought and extreme events. Please feel free to check out some of our other videos to learn about other ecological and physiological aspects of our work in more detail.